Imagine a world where a single treatment could stop not just one virus, but dozens, even those we haven't even discovered yet. Sounds like science fiction? It might not be for long. Researchers at the CUNY Graduate Center's Advanced Science Research Center, CUNY ASRC, have just unveiled a groundbreaking discovery that could revolutionize how we fight viral outbreaks. Their work, published in Science Advances, introduces a novel strategy to combat a staggering array of viruses, from Ebola to SARS-CoV-2. Using a previously overlooked target, viral envelope glycans. Here's where it gets really interesting. This approach isn't just a scientific leap. It's a potential game changer for global health preparedness. And this is the part most people miss. Unlike antibiotics, which can be broadly deployed against bacterial infections, while doctors identify the exact pathogen, antivirals today are often too narrow in scope. If a new virus emerges, we're left with limited tools until vaccines and therapies are developed, a gap that leaves populations vulnerable for years. So how did the CUNY team bridge this divide? By focusing on a shared weakness across viruses. Viral envelope glycans, sugar-like molecules on the virus's surface, are structurally similar even in unrelated viral families. These glycans act like shields, helping viruses evade the immune system and infiltrate host cells. Until now, scientists hadn't tapped into this common feature as a drug target. The researchers screened 57 synthetic carbohydrate receptors, SCRs, tiny molecules engineered to bind to these glycans. 4 standout compounds emerged, capable of blocking infection from 7 viruses spanning 5 distinct families. Think about that. A single treatment neutralizing Ebola, Marburg, Nipah, Hendra, SARS-CoV-1, and SARS-CoV-2. In a critical test, one of these SCRs was administered to mice infected with SARS-CoV-2. The result? 90% survival in the treated group compared to 0 in the control group. The mechanism? The SCRs latch onto the glycans, disrupting the virus's ability to enter cells. A process that's both elegant and unprecedented. What makes this even more exciting is its potential beyond viruses. The same binding strategy could be adapted for cancer therapies or immune disorders, opening doors to entirely new medical applications. Dr. Adam Braunschweig, the study's lead investigator, calls this a first line of defense against emerging threats. And he's not wrong. If a novel virus, like the hypothetical X virus, appeared tomorrow, we'd currently have no immediate countermeasure. These compounds could change that. But here's a question to spark debate. Could such a broad-spectrum approach lead to unintended consequences, like resistance evolving faster than with targeted treatments? Or might it inadvertently disrupt the body's own glycan systems? We'd love to hear your thoughts in the comments. Do you think this is a leap forward or a risky gamble? The next phase for the team? Pushing these compounds into clinical trials. With the global threat of pandemics looming, this research might just redefine our arsenal. For now, the full study is available in Science Advances, DOI 10.1126, Sci-Adv AD3554, and the original article can be found here. Let's keep the conversation going. What do you see as the biggest hurdle in bringing this to market? Share your views below.